There you are.
Hello.
Hi. I didn't think I could.
Can I ask you a quick question before school
Sure.
starts? Okay. Did you, so when I submitted my practical,
Yes.
it made me cut add it as like an attachment. Is that how I was supposed to do it? Because like on my thing in grades, it says that it's missing. And
Okay.
like,
So I haven't returned those yet, but I actually wrote you a little message on those. When you like for the next practical, there is a file that you have to open to see the questions. And that the reason I do that is because I often add pictures and things into practicals. But you can type your answers right into the quiz on Canvas. So you typed your answers on the file, right?
mhm.
And then you submitted the file. That's why Canvas doesn't think you did it. Because
Okay.
Canvas was looking for you to type the answers into the quiz spaces. The quiz.
Okay.
Yeah,
But
that's all.
you did get
Yeah,
it,
I got
right?
it. Yeah, yeah,
Okay, because
no
I was like, now I'm worried.
no problem. But yeah,
Okay.
that's what that is. I know
Okay.
it's a little strange, but because it's a little bit hard to embed images into a Canvas quiz, what we do instead is we use a separate file for you to open up and see the actual questions. So
Okay.
no problem there.
Thank you.
No problem. So it looks like we're just about all here. Still waiting for, I think, one other person. If you would just type your name into the chat. I see a couple of you have already done that. Thank you. Yeah, just type your name in for me. As usual. And we can get started. So I have graded your lab practical exam. There's still one student, though, who is going to be taking that exam. In fact, she's taking it today. So I will release those grades, as we say in Canvas speak. I will release those grades to you tomorrow once I have hers in and I have it graded. Everybody's grades will be returned back. Overall, though, they look very good, very, very good. So I'm really pleased. So I graded the first lecture exam, and of course, those grades were submitted as well. I was very happy with that first lecture exam. You all did really well on that. The average came in at a 94, I think. It was 75 out of 80. So that's very good for that first exam. Good job. When you go in and look at your lecture exam, you'll be able to see my comments if you did miss anything. Remember, you can click on that little link that says view. I think it says view instructor comments or something. You can click on that and you can actually see where I've written things underneath certain questions that you might have missed. All right, I'm just double checking to see who's here. Very good. Yeah, if you're coming in, just remember to type your name into the chat box. Vicky, are you here? I don't see Vicky.
The second one down, but I also typed in owner just to be funny.
Hold on, I found you. Okay.
I'm in there twice.
Oh, that's great. All right, good. We're going to go ahead and get started then, because I think just about everybody's here now. So this week, today, we're going to be talking about how we go about looking at microbes in environmental samples. So kind of an unusual topic. We talk a lot more about how we do things in medically related samples, but today it'll be more about the environment. We'll also talk a bit about culture media today, particularly about agar. and why we like agar and why we use it in the lab. Looking ahead, let me see if I can pull this up for us. And I'll share my screen with you so you can see it. Okay. So I've just pulled up our modules page in Canvas. Hopefully you can see that on your screen. So we're down here now, all the way down here. So you took your lab practical exam for me, and we'll be doing this lab today, microbes in the environment, and you will have some homework questions to answer and submit to me by Sunday. And then looking ahead to next week, we will not meet on Monday for lab because it's President's Day, it's a holiday. So that'll be good. You'll have that time to do whatever's most useful to you. And then on Wednesday next week is your next lecture exam. They come up fast, don't they? So when we go back, if you go back in our schedule, from the time since we took the first lecture exam, we've done prokaryotes. We've done, again, just lecture here, eukaryotes, and acellular microbes. So next week, that lecture exam will cover those three topics: prokaryotic cells, eukaryotic cells, and the acellular microbes and particles, the prions and the viruses. So that'll be on Wednesday. And this lecture exam will be very similar to that first lecture exam you did for me in terms of its format and its length. And you'll be able to take it whenever it's convenient for you during the day. Again, sometime between 8 a.m. and 8 p.m. We will then have a laboratory on Wednesday afternoon, and we'll be talking about the fungi on Wednesday. It's kind of a fun lab. It's I will say, people, you can take an entire semester long course about the fungi. They're so complex, a group of organisms, but we'll just talk about them for one afternoon. So, I'll try to make it interesting for you. All right, so I'm just rearranging things here. So that's what's coming up for us. And let me ask before we start the lab exercise today, if anybody has any questions about any of the lecture material or anything that you've been looking at, watching, reading, anything like that, anything you need to ask about that, this is a good time for us to talk. If not, what I'll do is just go ahead and get us started into the lab. So you should see a title screen on your screen right now that says Lab Microbes in the Environment. And again, this is kind of a fun lab, I think. It's an interesting topic because a lot of what we talk about is related to medicine and the human body and things like that. And today we're talking more about environmental microbiology. So we'll talk about testing methods, essentially, how we go out and get samples, how we handle those samples and so on, and what we do with them when we get back into the laboratory. And as I said, we're going to talk about culture media today, specifically about agar. We're going to talk about why agar is a good choice for us to use as a solid culture media. We're going to look at the way we use agar. We put it, remember, into Petri dishes to make these plates that we can grow bacteria on. We also use it in what are called agar slants and agar deeps. And we'll take a look at those and how we use those. And then we're going to spend the last part of the lab looking at the way bacteria grow on solid and liquid media. So we'll take a look at what colonies look like. That's what we refer to bacterial cells as they grow on solid agar. We'll look at how they grow, and we'll talk a little bit about what's known as their morphology. Colony morphology tells us what colonies look like. And as you might imagine, different bacterial species grow into different looking colonies on agar. So we'll take a look and we'll talk about how we can use that information about their look, their physical appearance when they grow, when we're trying to figure out what kind of a microbe we're dealing with. This word morphology will show up again and again through the semester. This just means body form. So anytime someone talks about morphology, they're talking about the form that an organism takes, the physical form. So we'll be talking about colony morphology. In other words, what do bacteria look like? What's their body form when they grow on solid media? So a little bit of background or review, I guess, is better. Remember that microbes are everywhere. They are all over us. They are all within us. They are in our environment. They are everywhere. Don't forget, most of them are completely harmless. They are not going to bother you if you come in contact with them. And in fact, many of them are absolutely necessary for your good health. So you're going to come in contact with them. We consume microbes almost constantly because we put our hands in our mouth. We put naturally grown foods in our mouth, and we breathe them in. We get them into our eyes. We get them into our urogenital tracts. We get them in and on us all day, every day. And of course, most of us are walking around very healthy. And that's because most of these microbes don't harm us. It's only a relatively small number of microbial organisms that are pathogens that are going to cause disease. Now, having said that, we do have to monitor our environment around us to make sure that whatever's growing around us is not a pathogen. For example, we talked about how we have to keep our recreational bodies of water safe for people to swim in and boat in and so on. And so states have to monitor the water and make sure that no pathogenic microbes are growing out of control. Water treatment plants, public water systems have to do this. If you buy a piece of property in a town and you are on the public water system, you get a report a couple times a year from your water authority telling you what the testing showed that they did and telling you, were there microbes growing in our water? And if there were, what were they and how many of these colonies did we find when we sampled the water? You can also imagine that there are places like hospitals and medical offices that also need to monitor, and especially monitor because those are places where sick people go. So it's super important that hospitals, number one, stay very clean, and number two, monitor to make sure that they're not growing pathogens in the little cracks and crevices and dark corners of the hospital. I know the majority of you are interested in healthcare as a profession, and you may be aware of the fact that hospitals have a real problem with the growth of pathogenic microbes. There are lots of cases where people, for example, go into the hospital for one thing and catch a pathogenic infection while they're there. That's not uncommon. People can go in sometimes for surgical procedures, elective surgical procedures and come out with an infection that they did not go in with. And that's because sick people go to hospitals and pathogens get spread. And despite sometimes their very best efforts to keep the place very clean, sometimes pathogens grow in those deep, dark corners of the hospital, and they can be transmitted to patients. So hospitals, medical offices, long-term care facilities have to be especially dedicated to monitoring the environment for the growth of pathogenic microbes so they can change up their practices if needed, in terms of disinfecting and cleaning and trying to get rid of those microbes. And there are even industries. There are many industries that maintain what are called clean spaces. I think I mentioned this early on in the course. There are these clean rooms in industrial spaces where they cannot afford for even a single microbial cell to get into the air in that room because it will damage or destroy whatever product is being made in that room. So they too have to monitor their environment in there and make sure that nothing is growing. So there's lots of people that do this kind of routine monitoring. And there are people who do this as part of their profession. They are involved in the environmental monitoring program, and they do this kind of work, sampling these environments so they can look for pathogenic microbes. So the kinds of things that get surfaced, well, the things that get sampled are a lot of times hard surfaces. If you think about, for example, in a commercial building, I mentioned to you previously that we have to make sure that the ventilation systems are free from pathogenic microbes, because when the air is blown through those ventilation systems, if there were microbes growing in the duct system, they could be spread throughout the building. So you can see this picture down here. Someone is sampling this very disgusting and dirty air vent. I think if I saw that air vent in a building, I don't know if I would go in that building. But you could see this person is holding like a cotton swab and is taking a little sample of this disgusting material on the vent. Here's another person who is taking a sample from inside a drain. This happens to be a floor drain. And again, in hospital settings, floor drains are very common, and they can get pretty disgusting. Water is used to wash the floor, and then that water is just sent down into the drain. And you would think, well, what does it matter if pathogens are growing in the drain? I mean, they're not going to spread to our patients from the drain. No, they aren't. But if somebody were to, for some reason, the cleaning staff, somebody had to reach into the drain, they might get those pathogens growing on their hands or on their gloved hands. The other thing is, this is just a good place to monitor, because if you know, for example, that let's say your hospital has a problem with an organism called Pseudomonas. If you're not familiar with Pseudomonas, it's typically not pathogenic to people who are healthy. But if you have a weakened immune system, or perhaps you're a burn patient, Pseudomonas can get into your body and cause severe, even deadly infections. So sometimes what a hospital will do is take a sample of the floor drain looking for Pseudomonas. And that's because what they're thinking is, if there was any Pseudomonas in this room, when the floor got washed, the Pseudomonas got sent down the drain. So I might find it in here. In other words, they don't want any Pseudomonas in that room. So if they find it in the drain, it means it was washed into the drain from the room, and they need to do something. They need to step up their cleaning procedures. We sample all kinds of things, solid surfaces in hospitals and doctors' offices, patient trays, chairs, the beds, the adjustable beds, the mattresses, We sample in the bathrooms, we sample the floors, we sample the stationary medical equipment. All kinds of samples are taken. So solid surfaces are very commonly sampled. And as you can see in the pictures, at least a couple of them, what we typically do is we use some kind of a swab. These are commercially available and they come sterile. So for example, you can see this person here at the top is holding onto a cap that has a swab attached to it. So this came in a tube, and the inside of the tube was sterile when it arrived. So this person just took the cap off and the swab comes with it, nice and sterile. And you can then sample whatever you need to sample by just rubbing that swab across the surface. If there are microbes there, you're going to pick them up on that swab. Now, that swab is then used back in the laboratory to inoculate some kind of media, whether it's liquid broth or a solid agar plate, it depends, but it will be used to, we will, in other words, try to grow on the media anything that might have been picked up on that swab. The other things we can do in terms of sampling, we can filter sample. A lot of times, instead of swabbing these ventilation ducts, what we'll do is we'll put a piece of filter paper inside the duct. We'll let the air blow through the filter paper, and then we'll use that filter paper to inoculate media back in the lab. And of course, the other thing we can do is collect liquid, and we do that a lot in terms of, again, things like lakes and ponds, things like water treatment facilities, things like public water systems. We're constantly sampling water. Now, that's going to be a very diluted sample. In other words, if there's microbes in there, there's going to be a lot more water than there is microbial cells. So those samples are almost always centrifuged first. They're put into the centrifuge and spun so that any cells will go to the bottom of the container. And then we will sample the bottom of that, the pellet, if you will, and use that to inoculate media. These swabs that you can buy, these commercial swabs, come in their own tube. And you can buy all different types of swabs depending on what you're sampling and how big you want the swab tip to be, and so on and so on. But an important thing to remember is this: you don't just take the swab and put it into a plastic bag and send it in to the microlab. You have to handle it very carefully. Remember, we've now used that sterile swab and we've rubbed it on a surface because we want to know if there are any microbes growing. So you have to now take this swab that has been used and you have to keep it from getting contaminated by anything else. So it has to go back into its carrying case. That's what this is. Not only will that carrying case keep it sterile, obviously, unless I've picked up some microbes, it will prevent it from getting contaminated with any other microbes on the way to the lab. But it also contains some media in it. It contains some liquid in it. And that liquid media has nutrients in it. Because if there are any microbes on the end of this swab, I want to keep them alive until I get them back to the lab so I can grow them and take a look at them. So you have to provide them with the things that living cells need, living microbes anyway. You have to give them some nutrients. We call that little bit of media in these carrying containers, we call that transport media. It's only enough to keep them going, keep them alive until they get to the lab. And then we're going to need to use this to inoculate some other media. So I've got a bunch of pictures on this slide, but these are just pictures of people sampling natural water sources. We have a very active water monitoring program here in New Hampshire for our recreational water bodies. You can see a couple people here in their fishing waders, and they're out in an area where people swim. You can see the buoy line right here, and they are taking samples. When you're a person who does this job as, as your work, it's often seasonal work in that they're going to hire you during the times of the year that that these recreational bodies of water are open to the public. But this is a fun job because you get to travel around from place to place and actually go into the water and gather samples. So not a bad way to make a few dollars. One thing to know about this kind of sampling, when you're going into a large body of water, like a lake, you can't take just one sample. So let's say I want to know what's growing in this little swimming area. I don't need to sample out here in the rest of the lake, but I do need to take multiple samples from the swimming area. I would want to take some samples from the shallow places and some samples from the deep places. I would want to make sure I got a nice representative collection of water samples from all around that swimming area. And the same would be true if I was working in a pond like this place, or even if I was at the seashore, and we do monitor at the seashore as well. You want to make sure you're getting not one sample, but several samples so that you're getting a representative collection of where people might get exposed to any microbes. People get very creative when they're sampling water bodies. This happens to be a municipal well. So this is where, this is the entrance down into this well that serves a community. And you can see they've attached like a jar onto the end of some string, and they're going to drop that down and get some water. You don't have to be, very fancy about this. You just need a container. These waters are not going to be sterile. They're going to have microbes growing in them. We just want to find out if they have any pathogenic microbes. And if they do have pathogens, we want to know how many cells that we can find per volume of water. So there's all kinds of plastic containers that they use. Each lab uses a different type of container. Again, it depends. And of course, those water samples just get carried back to the lab, and they would be centrifuged because, again, water samples tend to be very dilute. You don't have to worry about feeding those microbes with transport media on the way to the lab because they're in their natural environment. You have collected water that they live in, so you don't have to worry about them as long as you get them to the lab within a reasonable amount of time, which is usually half a day. As long as you get them to the lab within half a day, the cells are going to be fine in the water samples. Now, I put this slide in just more for interest than anything. These are handheld devices that you can buy. You don't need any kind of special licensing to buy one of these. These are devices that are capable of measuring microbial spores in the air. And there's all kinds of these for sale on the market. And they're heavily marketed to companies that are very popular now, by the way, that you can hire to come into your home and measure spores in the air. Now, you might ask why a person would do that. Well, there's a lot of fear in residences of fungal spores. There are a lot of people who are buying a brand new home, or perhaps have a person in the home who might be very sensitive to things like fungi, might be allergic to fungi. So they hire these companies to come in and use these meters to measure spore numbers in the air. Unfortunately, these are not super reliable. And let me tell you why. It's not that the instrument isn't reliable. This instrument can measure spores very well if it's used correctly. The problem is the instrument cannot tell the difference between a spore that is perfectly harmless. And again, I'm talking primarily about fungal spores here, not bacterial spores. It can count fungal spores very accurately, but it cannot tell the difference between a fungal spore that is perfectly harmless and a fungal spore that is potentially pathogenic. Remember what we said about spores. Spores are inanimate. They don't divide. They don't metabolize. They're not cellular. They're in a suspended animation. They have DNA in them, and the spore itself protects the DNA, and the spore will persist until it gets into conditions that are good. In other words, plenty of water, plenty of nutrients, plenty of warmth. When a spore gets into those conditions, it will revert out of a spore form and back into a cell form, what we call the vegetative stage or a cellular stage. So people don't like the idea that there are fungal spores in their house. But if you think about what a house is, it makes all kinds of sense that there are fungal spores. There are places in your home that are very, very dry. And it's good that they're dry. But if you're a fungal microbe and you're blowing around in this house, regardless of whether you're harmful or not, you're going to sporulate because it's too dry for you to grow. So it's very, very common to find fungal spores in residences that are perfectly clean, perfectly safe. Remember, microbes are everywhere, and fungi are microbes, at least some of them. So there are fungi in your home. Of course there are. There are fungi all over your body. So of course they're in your home. The problem is when they get off of you, they have to sporulate sometimes because they're too dry or they don't have enough nutrients or they're too cold. So these meters can be a little misleading because people hear that there are, let's say, 50,000 fungal spores per unit area, and they get afraid. They think, oh my gosh, the house is just full of fungus. But this device cannot tell us if those fungi are dangerous or not. The only way to know that is to get a sample and send it to the microbiology lab. And unfortunately, some of these companies that do this work don't take that step. Instead, they just report the number to the client, and the client then makes decisions about what they want to do. So it's unfortunate because it's a little bit of needless worry for a homeowner. Has anybody had any experience with this? Have you ever had a home that you bought or a home that you lived in tested for fungi or for spores? I joke about it a little bit, but there are some fungi that are very dangerous. You've probably heard of the infamous black mold that can grow in houses. That's a particular type of fungi called Stachybotrys. And while lots and lots of fungi are black when they grow, there is a particular type of black mold, Stachybotrys, that is very dangerous. It can make people very sick. So it's not that there are no fungal pathogens, there are. And if you have a mold problem in your home, if you have visible mold growing in rooms, you probably should get an evaluation done, but not just spore counting. We need to know what the species of fungus is so we know whether or not it's dangerous. All right.
I have a quick question about that.
Yeah.
Asking for a friend
Yeah.
If you see the mold is black, does that mean it's black mold?
No.
Thank you.
Most mold that grows in houses is black.
Okay.
Mold that grows on food can be black or green. I'm
Yeah.
sure we've all seen the green. Yeah. But black mold on food is very common. And so is different types of black molds in the house. Think about what grows in the corner of your bathtub, right? I mean, we call it mildew. We all call it mildew, but it's mold. And it's black and it's gross. But the reason it grows there is because your bathtub is often wet, right? And it's warm. It's a warm place. And that's why they grow there. So it's not that black means pathogenic with mold. It doesn't. But see, the average person doesn't know that. Some people have heard about black mold and they get very scared when they see anything black growing. If you imagine it, imagine if you walked into a bedroom and you saw some black mold growing on a wall, for example. It's not a place where you normally see it. So the fact that it's black, you might go, oh no, that must be the black mold I've heard about. In reality, it could be exactly the same mold that's growing in the bathroom. It's just that for some reason, you have too high a level of humidity in that bedroom right now. And the mold has started to grow. So I know it's gross to think about, but our homes have a lot of fungal spores in them. It's natural. The reason they don't tend to grow in most of our rooms is because they're too dry. Most of our rooms are too dry. Only the bathroom tends to be the place where we see them growing. Good question. All right. So we talked about how we can use sterile swabs to rub across a hard surface and perhaps collect some microbial cells. We said, we have to put those swabs back into their sterile containers. So there's a little transport medium around them to keep them alive for a while. And we said we can collect natural water samples just in containers. That'll keep them alive until they get to the lab. There's another technique that I want us to be familiar with because it's particularly common in hospitals. Sometimes, instead of swabbing a surface and then sending that swab in transport media to a microbiology lab, instead, what a hospital will do is use what's called a Rodak plate. A Rodak plate looks like this over on the right side. It looks like an Agar plate, except the plate itself, the plastic dish itself, is not the same. It's made out of a very flexible plastic. The Petri dishes that we use are not flexible at all. They're hard plastic. So this is like almost like a saucer of plastic. It has agar in it, and it's used in place of a swab. As you can see, what this person is doing, they have turned the Rodak plate over so that the agar is on the bottom. There's no lid on it right now. And they're pushing their finger down so that the agar touches the surface. Can you see that? So you literally press on the bottom of the plate and you will push the agar out just a little bit and you can press it down onto a surface. So you're essentially eliminating the middleman here. Instead of using a swab and then taking that swab and smearing it onto an agar plate, you're just taking the plate to the surface, essentially. You're rubbing your plate of agar onto a surface and seeing if you can pick anything up. Rodak plates have agar that has nutrients and things like that in it. So whatever they're picking up on this surface is going to grow right here on this plate. So environmental managers inside hospital systems will go around the hospital at intervals, maybe once a month or so, and they will sample surfaces with these Rodak plates. After you press this one plate right here to this one surface, you put the cover back on and you put it in an incubator and you see what grows. So again, it essentially takes out the middleman. You don't need the swab here. You just go straight to the agar dish. It's going to go in the incubator and we're going to see what grows on it. And then we'll interpret those. And I'll talk about that in just a minute. But, you know, not everybody uses Rodak plates. They are a little bit pricey. So in a lot of other situations, we use a swab. And when the swab gets back to the microbiology laboratory, the technician in the laboratory is just going to use a regular agar plate. That's what you're looking at here. Nice Petri dish, hard Petri dish with agar growing on it. And the technician is going to take that swab that was used to sample the surface somewhere, and it's going to spread it out over the surface of the agar. Now, I put these two plates in here because you can see there's some cloudy looking material growing on both plates, but it's growing very differently. Let's look at this plate on the left first. This is an agar plate. The agar, remember, is in the bottom part of the plate. We always keep these plates upside down because we don't want condensation to fall on the agar. Notice that, oh, there's a nice label on the side of the plate. That makes me happy. Somebody wrote their name and their date, and they wrote what kind of agars this is and what kind of sample they have streaked on it. Now, notice how it's been streaked. This is what we call a quadrant streak. Quadrant meaning 4 regions or 4 compartments. So what I want you to write down in your notes is this. A quadrant streak plate, and the words are written down at the bottom of your screen right now. The reason we make these is to grow individual colonies, and that's what you can see right here. Individual colonies of bacterial cells, so that we can examine them for colony morphology, or perhaps for additional testing. We could take a colony, for example, off of this plate, and we could run some tests on it. We could take a colony off of this plate and we could inoculate some broth with it. We could put it into a broth tube and subculture it. The point, though, is to grow these individual colonies. Now take a look at this plate. This plate looks very different, right? This plate is covered in colonies, spread evenly all over the surface. This plate is made very differently. This is called a spread plate, and it is used for the purposes of counting colonies. This one is just to get colonies to grow, isolated, individualized like this. And this one is to grow colonies in a way that we can count them and we can make sense of what that count means. So over here, this quadrant streak plate, I'll show you the way it's made. When you take your swab out of the transport container that it came in, the first thing you do is mentally divide this plate into 4 compartments. So imagine there's a line drawn here across the middle, and there's another line drawn here down the middle. So I've got 4 compartments here. The first thing you do is you go into the first compartment and you just smear that swab around. What you've done is you've put a lot of cells into that first compartment. And they're going to grow in a big, thick sort of mass of growth. And that's what this is. There's just a lot of cells growing here. And that's why it just looks like a big, just a collection, a smear of this beige-ish looking material. Now I'm going to take a sterile loop and I'm going to draw it across that first quadrant. I'm just going to slide it through across the agar like this. And I'm going to spread it out over here. And that's what this person did right here. You can see you've still got big streaks of growth here. You don't have individual colonies yet. But you have essentially taken a few cells from the first quadrant with your loop. You've dragged them into the second quadrant and you've spread them out. Then you take your loop and you draw through the second quadrant and you spread them out. And then you draw through your third quadrant and you spread them out. What you're doing when you make this kind of plate is you're getting fewer and fewer and fewer cells in each quadrant. You start with a lot of cells in quadrant 1, and hopefully you'll get individual circular colonies in quadrant 4. And that's what you're seeing here. Again, we don't do quadrant streak plates for counting purposes. We would not get a good count on this plate. We're just doing it to get isolated individual colonies to grow. This plate is different. On the spread plate, what we're going to do is we're going to take that swab and we are literally going to spread it all over this plate. In fact, what a lot of people will do is smear the swab first, then take one of those hockey sticks that I showed you and spread it all around. You're trying to get all the cells evenly spread over the entire surface of the plate. And when you do that, if things go well, you'll get individual colonies growing all over the surface of the plate. So quadrant streak plate for growing colonies, spread plate for counting colonies. So we can get a good idea of how contaminated a particular thing is by doing this environmental sampling. We can get an idea. We can do things like we can figure out how many cells there are per surface area. And typically it's things like centimeters squared. So for example, I could tell a hospital that, well, your floor in your cafeteria has this many cells per centimeter squared in it. So you need to step up your cleaning routine, something like that. We can also do it per unit of volume, say for a recreational body of water or maybe a well. That's typically per ml, per milliliter. And finally, we can do it by counting colonies. And we don't actually refer to them as colonies. We call them colony forming units or CFUs. So if I do one of these spread plates, I might report back that there are 300 colony forming units in the sample. Or maybe there are 36 colony forming units in the sample. And again, depending on where that sample came from, that number might mean, oh, we're doing a great job, or we have nothing to worry about. Or it might mean, oh my gosh, we have some work to do. We have a problem. It all depends on what it is that has been sampled. So I want to give us a definition for this word colony because I've been using it now for a minute or two. Each one of the little opaque sort of cream colored circles on this plate is a colony. Okay, so there are many, many, many colonies on this plate. All a colony is a visible mass of bacterial cells that are growing on that agar plate. Now, what's in that colony are clones. Now, let me explain what I mean by that. When I spread my sample out over this plate and I looked at it with my eyes, I didn't see anything, right? I won't see anything. I'll put it in the incubator, and any cells that are on that plate are going to start to grow. Now, within about 24 hours, the plate will look like this if it's got bacteria. Everywhere there's a colony that I can now see. What happened was a cell landed right in that spot. And that cell started to divide when I put the plate in the incubator. And for 24 hours, it divided and divided and divided and divided and divided until it made a little hill, a little mound of cells. They all came from the same starting cell, though. So they're clones. They are genetically identical to that original cell that landed on the plate right in that spot. And it seems like 24 hours isn't a very long time to go from one cell to a group of cells that is visible to the naked eye. But believe it or not, it's plenty of time. Most bacteria need somewhere in the area of 20 to 30 minutes to divide. In other words, that first cell that landed right here on the plate needs about 30 minutes to become 2 cells. Then after another 30 minutes, now we're going to have 4 cells clumped together, lying on top of each other. Then I'm going to get 8, then 16, then 32, then 64, 128, and so on and so on. It's exponential growth. So by 24 hours, that little mound, that little circle right there, literally has millions of bacteria in it. Each colony is actually millions of bacteria that all grew out of that one cell that landed there when I swabbed the plate. The reason we do this is because we can count colonies. We cannot easily count cells. So I cannot just look at that swab and know how many cells are on it. But if I spread it out onto an agar plate and put the plate in the incubator for 24 hours, now I can see something that I can count. I can see these colonies. And each colony came from one cell, theoretically. Does that make sense? Does that make sense to you? There's a reason that we make colonies grow. We make them grow because we can see them and we can count them. And I then know approximately, I'll explain in a minute, I then know approximately how many original cells there were on that swab that I smeared all over that plate. If I count the colonies, and I know that every colony grew as a big mound from one cell, now I know how many cells I started with. And I can report back. Yeah, that swab had 75 cells on it. Or that swab had 100 cells on it or 200 cells on it, according to this dish that I made here and I counted. So it's very, very useful. Colony forming unit counting is very useful for estimating how contaminated an environmental sample is, or how many microbes are in an environmental sample. All right.
I'm sorry, I'm going to ask to clarify.
Yeah.
Just a second. So you're starting with basically one parent cell in a sample, but maybe there's 2 because of how you inoculated it, is in one petri dish or one agar plate, for example.
So in your example, let's say I went into a hospital situation, because that's a good example, and I took one of my sterile swabs, and let's say I wanted to sample a drain. So I ran the swab inside the drain, and then I put it into its container and I sent it to the lab. And the technician took the swab out
Mm-hmm.
and rubbed it on the plate and got all the cells off of the swab onto the plate.
Yep.
If I grew 2 colonies on that plate, that means there were 2 cells on that swab.
Okay.
Does that make sense?
So you're you're counting the amount of colonies. So, but do you have to know the replication rate?
You actually don't. There are
I'm
bacteria.
sorry.
You actually don't. And I'll explain why. There are bacteria that divide very
Okay.
quickly. There are some bacteria that divide very quickly. And they're going to make visible colonies on that plate within about 18 hours. Some of them grow fast. There are other bacteria that need more like 48 hours to make a visible colony on a plate. Most of them take 24 hours. So what happens in a microbiology lab is if I come in, if I make a plate on Monday, if I smear a plate on Monday and I check it on Tuesday and I don't see anything growing on it, I just put it back in the incubator and I give it another 24 hours just in case. It just needs more time to grow.
Gotcha.
Sometimes you come
So
in
as
the
long as you
morning.
can count a colony,
Yeah,
it doesn't
as long as
matter
you've
what
got
time.
nice and right. Doesn't matter the size. Now, if I'm trying to do instead of just counting, if I'm trying to do colony morphology, if I'm trying to take a look at that colony and describe it, and I'll talk about that more in a few minutes, I might want that colony to be a nice big one. There may be colonies that are too small. But if I'm just counting, it doesn't matter how big the colony is. As long as I can see it, I can count it. I will tell you, there are some bacteria. There's a type of bacillus called Bacillus cereus. And this organism grows like crazy. And sometimes you'll make a plate on a Monday and you'll come back on a Tuesday and the whole plate will be like one giant colony. And it's because what happened was it grew so fast that the colonies just all melded together overnight. In that case, I would have to make a fresh plate and I would have to check on it a lot sooner, maybe 12 hours later instead of 24 hours later. So it all depends on the bacterium, but most bacteria that we end up dealing with in the micro lab take about 24 hours to form a visible colony that we can count. So
Great.
it's very
That helped.
handy.
That helped a lot. Thank you.
You're welcome. Okay. So we don't count, or we don't refer to colonies when we count them as colonies. We call them colony forming units, which seems a little strange, but here's the reason. Remember what a colony is. By definition, it's a mass of clones that arose from a single cell on a solid culture media. So there is a chance that some of those colonies arose from maybe 2 cells or 3 cells that all landed on the same spot on the agar. See what I'm saying? There's a chance that a single colony that I can see with my eyes didn't arise from 1 cell. Maybe 2 cells both landed on top of each other on that exact spot on the agar. So just in case that happened, in other words, to be technically correct, we don't refer to them as colonies when we're counting them. We refer to them as colony forming units because we're essentially going to use them as a cell count. I'm going to count up the colonies, and I'm going to turn around and say, well, there were 200 colonies, so there were 200 cells. And if they were all purely true colonies, they all came from one cell, I would be absolutely correct. But there's a chance that some of those colonies came from more than one cell. So we hedge our language a little bit and call them colony forming units instead. What we're doing is an indirect measure of how many cells were on that swab, or how many cells were in that water sample, or how many cells were in that ventilation grate. Okay, we're counting up colony forming units, and we're gonna treat them as though each one of them arose from one cell, but we're recognizing that ours is just an estimate. There may be some colonies that didn't come from one cell, but came from a small group of cells instead. So we're just trying to hedge our bet a little bit with this language. Okay, a colony forming unit is not exactly the same thing as the cell it grew from. It may have arose from 2 to 3 cells. So I don't want you to get too bogged down with that. I'm not going to ask you to explain why we call them colony forming units when we count them. I just want you to understand that we use colony forming units as an estimate of how many cells were in that sample. Any other questions? So when I make my count and I do my report in the laboratory, if I count up 175 colonies on my plate, I'm going to report that I counted 175 colony forming units. And that will be interpreted as that there were approximately that many cells on that plate when I started. Now, it's important to understand that a little bit of math has to be done, because I cannot say, for example, let's say I draw up 4 ounces of water out of a well. I need to be able to saying to a person, there were 175 cells in this 4 ounces of your well water. That's not really going to help them. I need to then do a little math, and I need to tell them how many cells per ml there are, or how many cells per liter there are. That's a much more usable piece of information for them. So we do a little more math than just counting the colony forming units. We also do a little more math so that we can give a more usable report to whoever it is that ordered the sample be made. We're going to talk a little bit more about how we make spread plates later in the semester, but they're very commonly used when we do environmental samples, because spread plates are made in order so we can count colony forming units. It's the whole reason we make them. All right. Now, when it comes to spread plates, it can be easy to count the colony forming units, and it can be hard to count the colony forming units. It depends on how many there are, right? So a lot of this work is done by hand, especially in smaller laboratories. The person takes the plate, like this one over here on the right, and instead of counting all of these little dots, what we typically do is we draw quadrants on the plate again. So we'll draw a straight line down, and we'll draw a straight line across. And then we'll count up how many colony forming units are in one quadrant, and we'll multiply it by 4. So again, it's an estimate. It's not an exact count, but it's good enough. It's good enough information. So you can do it by hand. Lots of labs have technicians doing this by hand. But if you're working in a lab that does lots and lots and lots and lots of sample counts, it's not going to be efficient to have technicians doing it by hand. There are a couple other things you can do. Number one, you can use a semi-automated system like this one down at the bottom on the left. What you're looking at here, these are not expensive. They're very commonly found in micro labs. It's got a light source that has a grid on it, as you can see. So I would take my plate and I would put it right here. And the light is going to allow me to see this grid through the agar. And again, I don't need to count the entire plate, but I need to count some representative area on the plate and then multiply it so that I know how many are on the whole plate. Now, what you do with these semi-automated systems is you have a special stylus. And what you do is you touch the end of the stylus to each dot on the plate. So you're not actually counting them in your head. You just touch it very quickly to each dot. So I would go touch, 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 like that across the quadrant. And the little machine keeps a count. Every time the stylus tip touches the plate, it makes a count. So this is a nice system. It's just a little bit quicker than me literally counting these in my head. Now, in the biggest labs, the busiest labs that are counting colony forming units from environmental samples, they use a fully automated system. It's computer automated. So that's actually what's happening in this picture on the right. What happens is you put the plate underneath this camera that's attached to the computer program. The camera takes an image of the plate, and the software then counts the colonies. The nice thing about a fully automated plate counter like this one, a computer generated plate counter, is that they're very accurate, but they're also very expensive. So you're only going to find these fully automated systems in the largest labs. Most of the labs that I've ever worked in were research laboratories. I have worked in a state laboratory. I worked in a lab in Vermont that counted colonies from milk samples. So farmers would send milk samples into the lab from their dairy farms because they wanted to make sure that the milk was clean, that it didn't have any microbes growing in it. But most of the labs I've worked in, we did our counts by hand. So I'm very fast at counting colony forming units. Now, what about these Rodak plates that I mentioned? Remember, these are basically very flexible agar plates. And you turn them upside down and you press the agar down onto a surface in order to sample for any microbes growing on that surface. Remember, they go into an incubator for 24 hours again, approximately. And then somebody evaluates the number of colonies growing on them. We sample with Rodak plates after the place has been cleaned. So the number of colony forming units is always going to be relatively small. Hopefully, you can see these numbers are quite small. 0 to 25 or 26 to 50 or 50 and over colony forming units on that plate, growing on that plate. How do I interpret these? Well, the answer is it depends. It depends on where you took that sample. Let me show you what I mean by that. The Rodak plates that are used in healthcare facilities come with a standardized table that you use when you're interpreting the results. And this one is very typical that you might find with a kit of Rodak plates. You can see on the left-hand side, the left column shows all these different places in the hospital that might be sampled. So floors in the operating room, floors in the OB wing, the obstetrics and gynecology wing, floors in the nursery, tabletops, just in regions of the hospital. Then in the patient room, the floors in the patient room, the tables in the patient room, the bathrooms, other floors, other surfaces. So it all depends where you're sampling. And then you can see you've got columns that show good, fair, and poor. So let's say I took a Rodak plate into the nursery and I pressed it onto a countertop where the nurses do their work. Maybe they make up formula there, or maybe they write up patient records there. The nursery, the OR, and the OB wing all have the strictest standards. That probably makes sense. So if I grew, let's say I count 3 colony forming units on my Rodak plate, that's between 0 and 5. That's going to get a good rating. The general cleanliness of that countertop in the nursery would be rated as good. What if I counted 10 colonies? Well, that's fair. And if I count 30 colonies, that's poor. That means I am not doing a good enough job keeping that counter clean in the nursery. So as you can see, depending on where you are in the hospital, the standards are different. Now, I will tell you this, these tables come with the Rodak plates. They come in a kit. But there are a lot of hospitals that keep a stricter standard than this. There are some hospitals that if they get even 1 colony out of the operating room, I'm sorry, colony forming unit out of the operating room, it's not good enough. If somebody takes a Rodak plate and presses it onto the table, the operating table, and they get even 1 colony growing, that's going to be a problem. So some hospitals, despite what the company says, the Rodak Plate company, some hospitals keep their own very strict standards. And they would not consider 0 to 5 good. They would consider that poor. It all depends on where you are and where you work in. So let's put a summary here. We can count colony forming units in liquid samples quite accurately, frankly, by taking a little bit of that liquid, putting it onto an agar plate and spreading it all over the surface of the plate. Remember our hockey stick tool. We can get a nice accurate count for lakes and ponds and water treatment places and municipal water supplies and so on and so on. We can also sample hard surfaces, solid surfaces in the environment using sterile swabs and smearing those all over agar plates. Or we can skip the swab and we can use a Rodak plate instead. We can grow, in other words, colonies on our plates and count them as colony forming units. There is something we need to remember though, and that's that when we take a sample off of a surface, we can only apply the results to that location. In other words, if I sample that countertop in the nursery, it's the countertop that I can talk about. I can't talk about the floor. I can't talk about the infant bassinets. I can't talk about any of that. All I can talk about is the counter. So there are some limitations when we sample surfaces, and primarily with those Rodak plates. Typically, when people are swabbing solid surfaces, they tend to collect multiple samples. In other words, they tend to swab multiple places. Rodak plates, by their nature, you can only use in that one place. You press it against one place, and then you're done. So there's limitations to sampling solid surfaces, especially when you use a Rodak plate, because technically speaking, you can't really talk about the whole countertop with a Rodak plate. You can only talk about that spot you tested. Maybe you pressed the Rodak plate off in the corner of that desktop or that countertop. Maybe that corner is very dirty because nobody uses it, or it gathers dust, or you see what I'm saying? So if you're going to sample solid surfaces and you're going to use Rodak plates, it's best to take multiple samples. Multiple samples. In other words, don't use one Rodak plate on that countertop. Use 5 Rodak plates and try to get a nice representative sampling of that entire countertop. All right. Before I start talking about culture media, does anybody have any questions? I don't expect you to be experts at environmental sampling. The idea is just to learn a little bit about it so you have some understanding of why it's done and how it's done. Some of you, though, when you go out into your professional lives in the healthcare world, you may be asked to do this kind of environmental sampling, or you may have an opportunity to take on that extra work and hopefully get paid a little more for it. But it's very commonly done in healthcare because we have a real problem staying on top of the growth of pathogens in hospital settings. And it's a lot of hospitals are working so hard to control pathogen growth in the hospital environment. But you can imagine if your hospital brings the sickest people in, then it's going to be contaminated. And unfortunately, you have to work very, very hard to keep the pathogen numbers in the environment low. But I tell you, hospitals put a lot of time and money into sampling because it's in their best interest to know what's growing and where it's growing so that they can kill it. They can disinfect it and kill it. All right. In the time we have left, we're going to talk a little bit about culture media. If we don't finish with this today, we will we'll talk about it at a later time. I'll just tuck it into another lab exercise. But
What? Oh, can I ask a question real quick?
yeah.
What time is the meat till? Because the times on like the calendars are all different. Even for my other class, they don't even match like the times that. Like, is this class till 6 today or 5:30?
So this class is supposed to go from, well, I'll say my understanding of when it's supposed to go is from 4 to 5:30.
Okay.
But I know some people have it listed as going longer than that. If we were in the classroom, in other words, it would go from 4 to 5:30. So I try to stop at 5:30 just in case, people have that schedule and they need to go to work or something. I'll try to stop at 5:30.
Okay.
But yeah,
Thank
I
you.
know it's written different times in different places, so I don't really know why. All
Thank
right.
you.
You're welcome. All right. So we talk a lot about media in here. We talk about culture media, which is basically some kind of material that we're going to use to grow microbes. We're going to put microbes into the media or onto the media because we want them to grow. Okay. Now, if I'm doing environmental sampling, for example, I want those cells to grow. So I'm going to have to give them the things they need to grow. And I have a short list of those things right here. They have to have water. If a microbe dries out at all, it's going to either die or it's going to sporulate if it can. So you've got to give them a source of water. You've got to give them what we call a carbon source. So that means protein. I have a word next to that that says peptone. There's a lot of peptone used in microbe culture media. All peptone is protein that has been partially digested already. So partially broken apart, in other words. A lot of protein sources are just the molecules are too big for the microbes to use them. So they're partially digested commercially, and that's what we call peptone. The microbe is going to use the peptone or the protein as a source of carbon. In other words, it's going to use it to make other molecules. We also need to give them a source of energy. And when I use that word, I mean, we need to give them something that they can use for energy. They can use it to make that molecule called ATP, which is a molecule that cells use for energy. So the energy source that we need to provide is some kind of carbohydrate. We also have to give them various microminerals, things like a nitrogen source, a phosphorus source, a sulfur source. So this is just the most basic things that all microbes need: water, protein, carbohydrate, and a handful of microminerals. Now, some culture media is just that. I mean, it's a short list of ingredients, and we know exactly what's in the media. So we would call that kind of media chemically defined. In other words, we know exactly what's in it and in what quantities. It contains only known ingredients. But there are other media that are called complex media. And basically what these are is a basic chemically defined media that we then add some kind of substance to enrich it. Now, what kind of substances do we add? Things like blood, eggs, milk, certain meat extracts. These things are very, very, they're very nutritionally dense, but we don't know exactly what's in them. In other words, I can't tell you every single ingredient, every single molecule that's in blood. It's just not known. I can't tell you every single molecule that's in a chicken egg. It's just not known. So I can add these things to some basic media, and I might be able to coax certain microbes to grow by giving them these very nutritionally dense foods. But since I don't know exactly what's in it, I know it's blood, but I don't know what blood contains exactly. We call that a complex medium. It has some ingredients that I know exactly what they are, and it has some ingredients that I don't know exactly what they are. There are lots of general purpose, chemically defined media available to us. In this course, the ones you'll hear me talk about the most are called nutrient. There's a nutrient broth and there's a nutrient agar. Later on in the course, I'll show you the recipe for how we make them, but they are chemically defined. They have a very short list of very specific ingredients. Now, when it comes to complex media, we're going to make something basic and we're going to add one of those complex foods into it, like eggs or milk or blood or whatever. Now, I want to mention a few things about agar. Remember what agar is. Agar is actually a carbohydrate, and it comes from algae, a certain type of algae. It's referred to as red algae, and it's a certain species of red algae that we use. But remember, algae have carbohydrate in their cell walls, and the type of carbohydrate varies by species. So this particular species of red algae uses agar in its cell wall. So we grow up the red algae and then we harvest the agar. And that's what we use to make agar plates. So agar itself is just the carbohydrate. And what we do is we add things in. We add in the peptone. We add in some carbohydrate for energy. We add in the microminerals. All the agar is, in other words, is the surface for them to grow on. They're not going to use the agar for nutrition, in other words. We have to add in the things that they're going to use for nutrition. It turns out that very few microbes can digest agar. They can't use agar as a food. But that's actually a good thing because we can spread microbes on top of agar and we can let them grow on the agar and they won't eat it. They won't make it go away by eating it and digesting it. Agar makes a great solid media for a couple of reasons. Number one, like I just said, microbes aren't going to eat it. They're just going to grow on it. We can easily add nutrients to it for the microbes to use for food. And probably most importantly, it's a solid in the incubator. It's a solid at room temperature and it stays solid in the incubator. Imagine if it liquefied at incubator temperatures, we would be in trouble. We would lose our microbes into that soup. It stays a solid in the incubator though. It doesn't liquefy until it gets all the way up to 100 degrees Celsius. So in the incubator, which is typically about 35 to 37 degrees Celsius, it's going to stay a solid. So it's a very useful material to grow microbes on, which is why we use it. You can, when you heat it up and liquefy it, you can pour it very easily into your Petri dishes. And when it solidifies, it's going to make a nice, smooth, solid surface for our microbes to grow on. So it really is a really great product. And you can buy agar. You can buy it in a powdered granulated form. And you can add in your own nutrient recipe. Whatever it is you're trying to grow, you can add in whatever nutrients you want to add into it. A lot of labs make their own agar. They buy the agar granules, they heat it up until it turns into a liquid, and they add in whatever it is they want to add. They add protein, they might add glucose or something as the carbohydrate. They add in a little bit of nitrogen or a little phosphorus, and they make their own culture media. That's the best way, efficiency-wise, to do it because it's inexpensive. But some labs just buy their culture media, and there's nothing wrong with that. If you're a laboratory that's not doing that much microbiology, a lot of places will just buy pre-made agar plates. So we only have a couple more minutes, so I'm going to show you this slide and then I'll let you go. Most of the time when we use agar, we're using it in this kind of agar plate, like you can see in the picture at the top right. The agar gets poured into this Petri dish, it gets allowed to solidify, and then we spread our cells out on top. But it is possible to use agar in a test tube, and that's what they're showing us here. Over here on the far left, this is just a broth tube. There's a liquid broth in here. But these two have solid agar in them. I should say these three have solid agar in them. You can pour agar into a test tube, and then you can lean the test tube at an angle and let the agar solidify that way, and you'll get this. See, the agar has solidified at an angle, and you get this nice slope in here. So I can use this the same way I would use a plate. I can spread cells out on this slope of agar. This is called an agar slant. Solid agar in a test tube that is allowed to dry on a diagonal. Now, why would I want to do this? Well, I dry it on the diagonal because it gives me a lot of surface area in here. And typically, if I'm going to grow something on a slant, I'm not trying to grow colonies. I'm just growing. I'm just trying to get it to grow. It's going to be really hard to get colonies to grow because this, even though there's a fair amount of surface area in here, there's not the same amount of surface area as there is on a plate. So I might just smear onto my slant and just put it in the incubator and just try to get something to grow. And then I might take that material and put it on a plate. It's just another form of agar that we sometimes use if we're trying to grow microbes and we don't need to form individual colonies. The other form is this one. In this tube, we've poured the agar in, but we haven't slanted the tube. We let the tube dry. We let the agar dry in the upright position. I would never use the surface of this agar because it's just too small. And that's not what this is intended for anyway. This is called a deep. This is solid agar in a test tube. And the reason we make these is because we want to put microbial cells down deep into the agar. Sometimes microbes don't like to be near oxygen. We're going to talk about that at length later in the course. Some microbes find oxygen toxic. And if they're exposed to oxygen, they will not grow. So I can use a deep to put some cells way down here out of the oxygen and tempt them to grow. They will grow inside the agar if the conditions are right. So you might wonder how I get the cells down here to the bottom. What you do is you use something called an inoculating wire. It's just an inoculating loop that has had the loop cut off. So now it's just a wire, a straight wire. And what you do is you dip the wire into the material, the microbial material, and then you stab it into this deep. So you're depositing cells into the agar with your wire. Down here at the bottom of the slide, you can see two deeps that have been stabbed and put into the incubator. I want you to see that you can see the stab line in the tube on the right, but I can tell you that nothing is growing in here. Now, how do I know that? Because look at the tube on the left. There is something growing in here. What has happened, this stab line looks exactly the same as when I made it. The agar is still clear, and the stab line is the same size, the same diameter as it was when I made it. But in this tube, I can see that now that stab line has gotten much wider, and the agar looks much more cloudy. That's because the cells are growing. I've got cells growing out of the stab line and into the agar. So this is an organism that doesn't like oxygen. That's why it's growing so well without any oxygen. And finally, these pictures here, these are commercially available slants. A lot of labs make their own slants. They make their own agar, so they make their own slants. They can pour their own slants. But some laboratories prefer to buy them. So you can just see that the agar is sort of moving up the side of the tube diagonally. It was just solidified while the tube was held at an angle. So I have a nice oval surface of agar right here that I can smear some cells on, again, just to see if I can get them to grow. I'm not going to be able to do much else with a slant. So we've got plates for solid media, we've got slants, and we've got deeps. Here's the deep. And these are slants. So I can use that very useful material called agar in a couple of different containers in the lab. And again, we'll be talking about different testing and things like that through the semester. So you'll see these again. You'll see, we'll talk about plates again. We'll use slants, we'll use deeps. So you'll get to see them again. I just wanted to introduce them today. All right, so we covered a lot today. Does anybody have any questions I can answer now? You should be all set now to do your laboratory homework for the week. Remember, that's due by Sunday at midnight. Don't forget, next Monday, February 15th, is a holiday, so we're not going to meet for lab. And on Wednesday next week, you have your next lecture exam. We do meet for lab on Wednesday. We'll meet Wednesday afternoon, but you also have your second lecture exam on Wednesday. So make sure your notes are all in order between now and then. Okay? Questions? Anything?
Thank you.
All right, ladies, I'm going to let you go. Have a good evening.
Thank you.
Okay, stay in touch.
Thank, oh, she's gone.